It is just too small. As you have learned, the gender ratio of males to females is 1:1. But look at this family. What is the gender ratio of David's kids? 3:1? That's not close to 1:1. How about Stephen? His ratio is 0:3, which is even further from the expected 1:1. So, why is the observed ratio different from the expected ratio? If we combine the children from both families, the ratio becomes 3:4, still not quite 1:1, but closer. If you recall the results from our dear friend Gregor's series of experiments for the colour and appearance of seeds, the sample size was in the thousands, whereas for the flower and height experiments, the sample size was in the hundreds. We can see that for experiments with a sample size in the thousands, the ratio for their F2 generation is closer to 3:1 than for sample sizes in the hundreds. The larger the sample size, the closer the observed ratio should be to the expected ratio. So when the sample size is small, such as less than 1,000, the observed and expected ratios will unlikely be identical. In addition, the union of gametes is also based on chance and probability. This means that a random sperm will fertilize a randomly ovulated egg. So the combination of alleles an offspring will have is based on chance and probability. The outcome of each fertilization is also independent of the others. For example, regardless of your gender, your younger sibling will still have an equal chance of being either male or female. It is not the case that if you are a boy, your next sibling will be female. So even if the sample size is over 9,000, the observed ratio can still differ from the expected ratio. These two reasons are sufficient and should be your first line of defense against O-level questions. However, there are still possible reasons for ratios to differ, even when the sample size is large. One such reason is selection pressure. Factors that influence the chance of whether a set of genes, essentially an organism, are passed on, are known as selection pressures. If we look at the sex ratio in different countries, we will notice that at birth, the ratio of males to females is 1.07:1, meaning that for every 100 females, there are 107 males. When extrapolated to the whole world, the difference is quite large. If we arrange them according to those with the highest ratio of males to females, we will notice two large countries by population size that are the main contributing factors, China and India. In both countries, traditional societal norms have been more sexist, and with China having had the one-child policy in the past, it is not surprising that the ratio of males to females is even higher for the 15 to 24-year-old age group. In the past, mothers often had to undergo abortions if they conceived a female fetus. So, for some genotypes, they might not survive until birth, affecting the observed ratio. Closely related to this is detrimental alleles. An example is the allele that causes Huntington's disease. Huntington's disease causes nerve cells in the brain to decay over time, affecting a person's movement, thinking ability, and mental health. It is caused by a dominant allele. If we look at the probability of getting Huntington's, it is actually quite high due to it being a dominant allele. If one parent is homozygous dominant, even if the other parent is not a carrier of the allele, all the offspring would have the disease. If a parent is heterozygous, while the other does not have the disease, there is a 50% chance that each of their children will have Huntington's disease. If both parents are heterozygous, then the probability is 75%. It is only if both parents are unaffected that the probability is 0. Despite the high percentage, the disease only affects 0.0075% of the world population. Symptoms usually start after reproductive age, but some individuals who have a family history might choose not to reproduce to avoid burdening their offspring. So some genotypes negatively impact survival and reproductive success, causing the observed ratio to differ from the expected ratio, As mentioned, only 0.0075% of the world population carries this Huntington's allele. So even if it were to spread normally, the observed and expected ratios would still be different due to unequal starting numbers. Remember our blood groups. O is the recessive phenotype, while A and B are dominant phenotypes. AB is codominant. Instinctively, one might think that blood group O is the most uncommon blood group. However, when we look at the blood group distribution in Singapore, O is actually the most common blood group. The dominant blood groups A and B are nearly half that of O, while the codominant AB blood group is the least common. Looking at the distribution of blood groups worldwide, it is roughly the same, but there are about 5% more people with blood group A than B. Since originally there was only the O blood group and A and B only came about due to mutation, the majority of the population still has blood group O. In summary, the two most common reasons for differences between observed and expected ratios are small sample size, and the observed ratio is based on probability. Other possible reasons for these differences include selection pressure, where certain alleles or chromosomes are favoured, and the presence of a large portion of the population that still carries the original allele. So it must be noted that a dominant allele does not necessarily represent a beneficial phenotype, nor does it mean that most of the population has that phenotype. Dominant alleles simply mean that their phenotype will be expressed in both homozygous and heterozygous conditions.